Hello and welcome everyone to this podcast for cell biology. In this podcast, we're going to talk about cell division. And we're going to spend a lot of time focusing on one aspect of cell division called mitosis. Before we get started though, I wanted to spend just a little bit of time revisiting the cell theory. The cell theory can be described by these three tenets. The first is that all living organisms are made of one or more cells. So the second one is the cell is the basic unit of life. The last one is that cells arise from pre-existing cells. They don't just spontaneously form. You have to have a cell before you can create the next cell. And for this podcast, we're going to focus primarily on this last tenet. So basically what we're going to talk about is how we take one cell here and generate two cells. In order to do this, it doesn't just happen by accident. It's a organized, regulated sequence of events. In this process, two large things have to happen. The first is that material needs to be copied. We're going to focus primarily on genetic material, but don't forget that all the organelles in this cell also need to be, but don't forget that when you make two new cells, you also have to make sure that they each have a proper complement of organelles and cytoplasmic factors. So those also have to increase before the cell divides. But we're going to focus on the DNA, the genetic material. After this genetic material is copied, it needs to be segregated. So we copy it, and then we need to make sure it gets properly segregated into the new cells. Okay, so when we think about this cell division, we can think about it as existing in three types. The first is binary fission. Binary fission is seen in bacteria and archaea. In eukaryotes, there are two types of cell division: mitosis and meiosis. So they both are found in eukaryotes and not in bacteria. We're going to spend time in this class talking about mitosis. You'll learn about meiosis when you take genetics. But for now, let's highlight the main difference between mitosis and meiosis. Mitosis, its purpose is to make genetic clones of the original cell. When you go from this cell to two new cells, these two new cells are genetically identical to this cell here. Most cell division within eukaryotes occurs by mitosis. So what is the difference between mitosis and meiosis? In meiosis, when we make new cells, we generate genetic diversity. When this cell is copied, the two or four resulting cells are genetically distinct from the original cell. The cells that are produced by meiosis are gametes. These gametes could be sperm, egg, they could be spores, but this is important in sexual reproduction. And I'm almost out of room, so I apologize for writing so small down here. I'll repeat this in case any of this gets caught off. Gametes are our sperm, eggs, or spores, and they're important in sexual reproduction. And you'll learn about all of this when you take genetics. Now I want to spend just a little bit of time on the side here talking about the time of division. And I'm only going to, again, for the rest of this podcast, talk about mitosis. And you'll see that the timing of cell division varies quite a bit depending on what type of cell you're talking about. For instance, an embryonic cell, they divide really fast. In humans, it's about 30 minutes. A yeast cell, similar to what you studied in lab, it will take between 1.5 to 3 hours. to divide. A human fibroblast cell, these would be cells that would make our collagen. So they would be connective tissues. They divide every 20 hours or so. So they take a little longer to divide. Our intestinal cells, they don't have quite as long of a lifespan. We lose them quite frequently. So they have to divide a little faster. For them, it's about 12 hours. And then we have some cells like neurons, and they don't divide. Once they're made, they're there forever. So if you lose them, you can't get them back. So they don't divide. Okay, now let's move on. And I want to talk more specifically about those two steps. Copying the genetic material and then segregating the genetic material. And to do that, let's talk about the cell cycle. We can think of the cell cycle as occurring in two distinct stages. Up here is the M phase. This is mitosis. M phase takes about 1 hour. And this is the part of the cell cycle that involves segregating the genetic material. So how do we copy the genetic material? Well, we copy the genetic material in this other part of the cell cycle. And this other part is called interphase. Interphase, depending on the cell type, as you saw on the last board, but it can be as much as 20 hours. This is the part of the cell cycle where the genetic material is copied. Interphase can be divided into three distinct parts: G1, G2, and S phase. So let's talk about these three parts of interphase. So this first part of interphase we will call G1. This stands for gap 1. This next part, this is called S phase. S stands for synthesis. So this is the part where the DNA is going to be copied. And then this last part of interphase is called G2, which stands for gap 2. The gaps are what separates the S phase between the M phase. So let's talk a little bit about what happens during these phases. In gap 1, the cell will begin to grow, get a little bigger, and during this time, the cell will monitor the environment. Is the cell getting a signal to divide? Is it getting a signal to not divide? If it's getting a signal to divide, then it will move on to S phase. But if it doesn't get that signal, then it won't move on to S phase. We'll talk more about this later, but during this time it will also check for mutations. And if there are mutations, it will either fix the mutations or it will kill the cell. I should also mention that G1 lasts, depending on the cell type, but if we think about a 20-hour cell division, approximately 20-hour cell division, this lasts about 11 hours. During S phase, as I already mentioned, this is when DNA is replicated. It makes an exact copy of the DNA. S phase, depending on the cell type, will last between 6 and 8 hours. So let's talk a little bit about G2, our second gap. Pretty much does the same thing that happens in gap 1. So let's repeat them. Cell grows. This is important because after you've copied the DNA, and as you enter M phase, you're committing to making two separate copies. So you need to make more of that cell membrane so that you can have two cells emerge from M phase that are about the same size. Also, the cell will monitor the environment. It also checks for mutations and verifies that all DNA is copied. G2 lasts about 4 hours. Okay, and we're going to come back to M phase later when we talk in more detail about mitosis. But as I said here, the DNA during M phase is segregated into two new cells so that each cell has a copy of the genetic material of the DNA. Okay, there's one more part of this cell cycle that we need to include, and it's right here. And it's called cytokinesis. Cytokinesis occurs after that genetic material has been segregated to each of the new cells. Cytokinesis pinches off each of the two new cells into distinct cells. Cytokinesis is responsible for generating this final step of making the two new cells. Now I want to just spend just a little bit of time talking about the timing of these events. What I described here are the typical times for a fibroblast cell. But if you remember on the previous board, I said that an embryo divides much quicker. In fact, if you were to look at a newly fertilized egg, what we call a zygote, and then you look later at a 16 cell embryo, and then even later at a stage called a blastocyst, which I'm not going to ask you about on a test or a quiz, but I'll write it down here anyhow, where that embryo has started to form a little bit here, the size is not that much difference. In fact, between a zygote and a 16 cell embryo, the size is almost the exact same. And as you move up to the blastocyst, it is a little bit bigger. I should make it a little bit bigger, but it's not much bigger. And the way that happens, and the way that you speed up cell division so that this cycle occurs much faster for an embryo, is that you essentially eliminate G1 and G2. Still there, but it's very, very short. S phase gets shortened a little bit, as does M phase, but the way you really shorten this whole cycle for an embryonic cell is to remove G1 and G2. Okay, so, alright, I'm going to redraw the cell cycle here that we just talked about, where we had M phase up here, we had our two gap phases, G1 and G2 here, and then we had our S phase here. As we said in the very beginning, the purpose of this whole cell cycle is to copy the genetic material and then to segregate it into new cells. So I think it's appropriate now that we spend a little bit of time talking about that genetic material that we're going to copy and segregate. All that genetic material in the nucleus is found on chromosomes. And a chromosome may look something like this. On the ends here we have something called telomeres, which you'll learn more about in genetics. They're important in maintaining how long this chromosome will exist. In the middle here, we have a centromere. This is going to be important for today's discussion on mitosis. The centromere, I drew it in the middle, but depending on the type of chromosome you're talking about, it can be anywhere along the entire length of the chromosome. But for our purposes, I'm just drawing it in the middle here. Now the chromosome itself, the whole chromosome, including the telomeres and the centromere, is composed of DNA. In fact, this whole chromosome is nothing more than one long piece of DNA, one giant double helix. Every chromosome is one giant double helix. Now what holds this together are proteins. There's lots of proteins involved here, but I would like you to know the word histone. There are lots of different histones that can hold this DNA together, but I just want you to know the word histone as being one of these proteins necessary to hold or to compact the DNA. And you'll learn more about histones when you take genetics and how they're important in regulating which genes get turned on. These are the two main parts of the chromosome. We will of course also see RNA here as well, but it's not typically considered one of the main parts of chromosomes. But RNA will be found along the chromosome, involved in various aspects of replicating the DNA and transcribing the DNA and regulating the DNA. Now, a little bit more about the DNA here on the chromosome. There are segments here, and many more than I'm drawing here. This is just for simplicity. In this case, I drew, 5 here. And each one of these are different genes. So gene 1, gene 2, gene 3, gene 4, gene 5. A typical human chromosome would have thousands of genes on them. Each of these genes, as we've talked about before, and you'll learn in more detail when you take genetics, is responsible for making a different gene product. That is, making a different protein. So for the cell or the organism to continue to exist, it's critical that these chromosomes, which contain the genetic material, are copied, and that new cells have an exact replicate of them, so that when my liver cells divide, they produce liver cells that will behave as liver cells. So what I want to do now is talk about the chromosome as it's going from G1 to S to G2 to M phase. And I will introduce a few other terms as we go through this process. I accidentally erased the cell cycle, so I'm going to draw it again here. With our M phase, our G1, our G2, and then our S phase. Now in the last whiteboard, I talked about the chromosome and how we were going to talk about the chromosome as it's being copied and it gets segregated. But I want to say a couple more things about chromosomes before we move forward. On the last slide, I just drew one chromosome. But in most animals, such as humans, for every chromosome we have a second chromosome that has the same genes on each of these chromosomes in the same order. So we would have gene 1, gene 2, gene 3, gene 4, and the same genes over here. There could be mutations between the two of them, but essentially they're the same chromosome with the same genes on them. We call these two chromosomes homologous chromosomes. And remember, in an organism, say like humans, we would have 23 pairs of chromosomes, each pair being a homologous set of chromosomes. So we'd have a total of 46 chromosomes. So for instance, our next chromosome, say if this is chromosome 1, this would be chromosome 2, we would have another homologous chromosome. And then chromosome 3 here, like so. Each set of homologous chromosomes would have a different set of genes on them, but together these would all be homologous chromosomes. Now as we talk about the chromosomes going through the cell cycle and getting segregated, we're only going to talk about one chromosome in a cell. So if I draw a cell here, I'm only going to draw this one pair of chromosomes, this one homologous pair of chromosomes. Now in G1, before they go to S, the cell gets a little bit bigger. So in S, I'm going to draw the cell a little bit bigger. And during S phase, these chromosomes will copy. And I'm going to draw it in the same color here, and I'll explain why in a moment. This orange one on the left is this orange one here. This brown one here is this brown one here. The one I drew next to it is the copied one. When it copies, we call these two chromosomes together here, we call them sister chromatids. These two are also sister chromatids. Don't get confused with homologous chromosomes and sister chromatids. That's something that often gets confused. During S phase, the homologous chromosomes are copied. The copied pairs here are called sister chromatids. The sister chromatids are identical to each other. Remember over here I said they're similar in that sense that they have the same order of genes, but there could be slight differences in mutations between these two. But over here, they're exact copies. They're identical to each other. The two orange are identical, the two brown are identical. I didn't draw them before, but I'm going to just draw a centromere on these right now. They're connected at the centromere, but they're also held together by a different protein that I'm going to talk about just in real general sense. I'm going to draw some like dots here between the two sister chromatids. And these dots are a protein called cohesin. Cohesin is a protein that holds the two sister chromatids together. It'll start to break down as we enter M phase, and we'll talk about that in a moment, but it's important that they're held together at this stage. Now, as the cell is completing the process of copying the DNA, it will enter G2. G2, the cell will even get a little bit bigger as it prepares for M phase. At this point here, we still have our two sister chromatids like so. Still with their centromeres, that doesn't change, and still with the cohesins holding them together. The two sister chromatids still held together by the cohesin proteins. Now what's going to happen in M phase, and we'll go in much greater detail in just a moment about what happens in M phase, but the result is we're going to end up pulling these sister chromatids apart so that over here at the end of M phase, at cytokinesis, we'll end up with two cells. Each one of these cells will have a copy of this homologous chromosome pair, just like we started with here. Okay, I think this is a convenient place to stop for this podcast. We've taken our cells through G1, through S, and now G2. In the next podcast, we're going to focus on what's happening here during M phase. So we've copied our DNA, and now we're going to segregate it on the next podcast. If you have any questions about the material in this podcast, please let me know. If not, I'll see you on the next podcast. Bye.